Thank you. So I'm really happy to be here, and I will try to talk a little bit about biological variation of blood EVs. So I've updated the title a little bit to be more correct. Okay, first, a little bit about my home country. Because it's such a small country, I have to promote it a little bit. And you can see it on the map. It's in the South Central Europe. And you can see that there is a lot of beautiful nature and beautiful sites to visit. But we also have a very enthusiastic and active network of Slovenian EV researchers. And if you're interested, please check here. But now to the important topic, to the biological variation. So this is a phenomenon where variability of laboratory tests results within a person or between individuals. And if we take a very simple example of, let's say, human height, you can see that in this left picture, you can see that during our lifetime, our height changes. So we could say that there is a random variation or there is a variation around a subject mean, around this homeostatic set point for each individual. But then if we compare these individuals' homeostatic set points in a larger population or cohort, then you can discuss, or you're actually looking at the inter-individual biological variation. And in here, there is a simple graph that represents the resolution or the distribution of heights of working females in Europe. And you can see that they're separated by sex. So females are smaller, have sizes from, let's say, 150 to 175, while males are larger. So we are here comparing different homeostatic set points. And how do we describe this mathematically? So we can express it as coefficient of variation, which is a simple calculation. So standard, it's a ratio between standard deviation and sample mean, and multiplied by 100 so that we get percentages. And if we have this normal distribution, this means that inside standard deviation around mean, there is 68% of all data would fall in this region. So we then also talk about individual coefficient of variation, and as I mentioned before, group coefficient of variation. There is a simple example presented here where we have three people who each of them have different, you can see, let's say for the orange one, there is a huge or at least larger coefficient of variation, and we can compare those and also calculate the group coefficient of variation. So for the biological variation, so for the blood parameters, you can appreciate from this diagram that this biological variation is inherent to those parameters. If we just look at a few examples, so for the glucose, fasting glucose, you can see that there is a range of possible or let's say the reference range of possible concentrations from a little bit below 4 to up to 6 millimole per liter. And then for the hemoglobin, again, you can see that this is dependent on sex. So females have lower hemoglobin reference ranges compared to the male hemoglobin levels. And this is known for the blood parameters, but for the extracellular vesicles, this is mostly unexplored, so there is almost, I would say, no literature on that. And why is it even important to understand better about this biological variation? Of course, first thing is we want to understand biology of circulating EVs better. So this is the fundamental knowledge that we want to increase. But then it's also important to define reference ranges for these EVs in normal or healthy population, so that it's much easier to detect what is significantly different in disease. And of course, it's also important to understand the sources of this variability so that we can account for that when we are building patient cohorts, and of course that we can also account for that in EV reference ranges. So our study will try to be the first attempt to evaluate those biological variation in circulating EVs. And I will just briefly introduce the cohort characteristics. So to that end, we enrolled 208 blood donors, and they had no known health issues or disease or infections, and also they didn't take any medication. So even if they only took hormonal therapy, they were excluded from the cohort. And then we collected demographic, clinical, and laboratory data. All were from South Slavic ethnicity, so Slovenians, from 18 to 65 years in age, and half were females and half were males. And then we collected general health data, body mass index, blood pressure, heart rate, smoking status. Okay, I'm not going to read all of this, but you see that we've tried to take a lot of information, and also laboratory data. So we've performed complete blood counts, hemoglobin levels, lipoproteins, then CRP levels, and insulin levels. And it's difficult to say this. I know that we could discuss this for the whole day, what is healthy, but for us, they all had all of the ranges, all of the parameters were in the reference ranges for that parameter, or were at least typical for Slovenian population. And that's why for us this was a healthy population. And then we collected blood samples from them and processed plasma. And again, I could talk probably for another half an hour about how we try to minimize all of the pre-analytical variables, but let's just say that we try to use standard protocol in the same facility. There was no transport. Everything was done quickly by the same person, and we've also done a lot of, or at least try to do some quality control of the blood plasma. And now to the study. So as I said, we try to we try to understand the circulating EVs better. So what we did was we phenotyped those EVs by imaging flow cytometry and by multiplex bead-based flow cytometry. So for the imaging flow cytometry, this was done in collaboration with Bernd Giebel's lab. So we've quantified EVs directly in plasma. And we've used 25 different markers that are typical cellular proteins or are indicative of a parental cell. And then for the multiplex bead-based flow cytometry, we first had to isolate EVs from plasma, and we've used a method we use in the lab a lot, so ultracentrifugation through sucrose cushion. And with these isolated EVs, we've performed this phenotyping again, in this case for 23 cellular markers. And what's important here is that because of the detection method, all of the EVs, or the population of the EVs that we've studied, is actually only tetraspanin positive subpopulation. So it's not the whole population of EVs, but just those who are tetraspanin positive. Okay, and let's just go directly to the results. So from the blood counts, we could quantify different blood cell types in our patients, or not patients, our healthy adults. And what we could see is normal for blood cells, that erythrocytes were in the range of, I think, about 4 to 5 to 10 to 9. And then for the platelets, they were 22 times lower in concentration, while the leukocytes were about 790 times lower in concentration compared to erythrocytes. And of course, there's a lot of complexity with leukocyte, but we try to at least focus on some markers for lymphocytes, monocytes, and granulocytes. And the sizes of the circles with this full line are indicating the concentration, while the shadows around this line indicate the CV, so the coefficient of variation. Okay, And from these patients, from these cells or from this blood, we've also, as I said, analyzed phenotyped EVs in plasma. And again, we use the similar representation, so the size of the circle with full line is indicative of concentration, and then the shadow is indicative of the CV for this specific EV subpopulation or subset. From these different sizes that you see here, you can immediately observe that there was a lot of different ranges of concentrations for different EV subsets. And what's even, what probably is even more obvious is that the shadow is huge, so everything looks colored. And this was actually what we noticed, that there was a huge individual variability in concentration of these subsets. I think that the median was about 160%. But actually, if you look at the data, you can see that some of them were up to 700% in CV, and then let's say others were, of course, much lower, around 70%. So there's a huge variability in concentrations of these specific EV subsets. And what was interesting, if we separated this by sex, we observed that at least in these 3 cases presented here, it seemed that, so for the CD90 and CD62P and also for CD235A, we could see that this variability is mostly on the account of the females. So there's a lot of variability in females that is introduced in the whole population. And of course, we were interested to learn more, so we've done complex association analysis, trying to understand what are the major sources of this biological variation. And first observation was that there's no global effect. So it's not that one parameter, one subject's parameter would affect the whole population of EVs in blood. But we could, so there was no global influence, and this is presented here in this principal component analysis, where with the input data were all of these different concentrations of EVs subset that we measured. And we could see that the population looks quite compact, so we couldn't see that there's something that would separate a population in 2 cohorts. And if we just colored this in different ways, so in this graph we colored this based on sex, you could also see, yes, everything is mixed. There's no real distinction based on sex. But importantly, we could see that sex did influence some of the EV subsets. So in this case, we could see that PAC1 positive EVs concentrations were higher in females compared to males. And this was not due to also changes in platelet concentrations or activation or something like that in females compared to males. So this seems specific just for EVs. And we've done similar stuff for menopausal status, and again, no global influence, but we could see that some leukocyte EV subsets were influenced by that. And in this case, you can see that postmenopausal women have lower concentration of CD60B positive EVs, or CD100 positive EVs, or CD152 positive EVs. So lower concentration in postmenopausal women. And actually, these were more similar concentrations to what we observe in men. So it seems that hormones, of course, have important effect. And again, we couldn't see the same thing for the whole blood count, in the whole blood count. So it seems it's not related just to more or less cells. And for smoking, we could see that smoking decreases CD235A positive EVs that are typical of erythrocytes. Again, this is not due to erythrocyte changes in concentration or hemolysis. So it does seem that there is some specific influence of these different subject parameters on specific EV subpopulations. And then, of course, we were also interested in these tetraspanin positive EVs. Just to remind you that here this phenotyping was done on isolated EVs that are positive for one of the 3 tetraspanins. And because there is no erythrocyte positive marker included in the assay, we couldn't show this data. And what we could see in this case was that, because here, of course, this is a semi-quantitative method, and what you actually do is measure or at least kind of understand what is the expression level of certain protein on the surface of this isolated tetraspanin positive population. And we could see that there's the highest expression, if we just look at the quantification, the highest expression level was found for the platelet specific markers, and it seems that these tetraspanin positive EVs are mostly from platelets, but there were also, of course, we could detect some from leukocytes and also from endothelial cells. And you can already see here that the shading is much thinner. So in this case, the coefficient of variation, so this biological variability, was smaller compared to what we've observed from the previous direct detection of EV subsets in plasma. And it was, the median was about 60% compared to 160 before. Yes, and of course there's still differences, so you can see that there's this CD62 positive EVs or expression has much higher higher CV compared to, let's say, CD9 or CD63 tetraspanins, or let's say even for the endothelial cell EVs have much lower CV. Okay. And when we've tried to understand the influence, what was the source of this biological variation, again, no global influence, but we could detect some specific influences of, let's say, age, and you will see later also sex on endothelial EVs. So if we look at CD105 positive, CD105 expression on these tetraspanin positive EVs, we could see that there is a difference between age groups. And this was similar to CD146 expression levels. Again, we could see difference between age groups, that there is significant difference. And this difference in expression was higher if we only focused on males. So this was even more significant in between the groups, the change. Okay, for the smoking, it actually affected several of the expression levels of different markers on tetraspanin positive EVs. So for the let's say those who are more connected to platelets, we could observe that there is an increase in expression level of CD63, of 41B, of CD62P, and then also CD9. So those two tetraspanins are more connected to the platelet EVs. But on the other hand, if you looked for the leukocyte EVs, we could see a decrease in concentration dependent on smoking in CD8 expression levels and in CD14 expression levels. So there is a complexity in response to the smoking in the circulating EVs. Okay. And if we just try to put all of this together in one scheme and also put our data into the context of what was published before, we can say that similar to what we've shown, Mobares et al. Also showed, so that CD41 positive, these microvesicles are influenced by smoking. And I should maybe point out that we've actually looked mostly at smaller EVs, and that other studies mostly looked at these larger EVs with flow cytometry. Okay, and then they also additionally showed that CD62P positive, these MVs are also influenced by smoking. And then similar to what we've done is that CD9 levels on tetraspanin positive EVs are dependent on smoking status. And then another study showed that, similar to ours, that CD105 positive MVs are influenced by age. And they also showed additionally by menopausal status. But there were also other studies showing that CD42A positive MVs their concentrations are larger in females, or that, let's say, CD235A positive EVs, so these erythrocyte EVs, are influenced by menopause and age, and that another study showed that CD41 positive MVs are influenced by age and smoking, and another one talked about CD62E positive EVs in females, and how, again, sex is the source of variation in these concentrations. So there seems to be some smaller, let's say, we are trying to build this knowledge. It seems to be we are getting slowly somewhere, and that mostly everyone showed that this influence is specific and it's not global. So, we have to have a big coffee ready because we still have a lot to do to understand this better. So of course, we didn't even touch on the intra-individual variation of circulating EVs. And we also we don't understand effects of disease of individual circulating EVs on this biological variation, or all of this needs to be studied further. And of course, also the sources of biological variability need to be studied. So nothing is known now connected to genetics, which is probably one of the most important influencers, and we don't understand it yet. And there, of course, in the end, only one study, I was getting worried when Ring presented his data, so please believe our data. So this is not only lipoproteins, but I think he got a really important point through that we have to then do inter-laboratory studies and trying to understand our data even better. So, and also use orthogonal methods. So I think he made a very important point. And in the end, it's also important to understand is it for EVs, will we be able to do population reference ranges, or will the personalized reference ranges be much more appropriate? Because as you've seen, let's say if the population range is very wide, and even if your, let's say, individual range, individual reference range is very small, even though there is a disease present and there is a difference that is actually showing that there is a pathology going on. If we use population reference ranges, this is then hidden in the whole interval if it's too big. So maybe for the EVs, individual reference ranges will be much more appropriate, but we don't understand this at all. So this is something that we need to study further. And in the end, just to acknowledge everyone that is here on the slide, especially Maria and Ivica, who are really instrumental in this study. So I'm happy to answer any questions you have. Thank you.
Sarah has a question. So we only have 1 or 2 minutes for questions and answers. So, can you use the microphone, please? It's coming. Wait.
Only 1 minute anymore. So excellent. I loved your presentation. Good that you mentioned the genetics behind. Coming from the blood service, we know that the very, very fixed and traditional measure that we do hemoglobin from the donator, that is it allowed to donate blood or not, is still like in evolution at the moment. So we are doing this individual evaluation currently, and that we know that the people has really different kind of recovery time. And we know that depending on the measuring winter time in Finland, or summer time in Finland, or morning time in Finland, or maybe somewhere else, and evening time, afternoon donors, that really has an impact even to the hemoglobin amount detected. So genetic variation, I think, is important, but then also this, I wanted to say, external factors that affect what the people has been eating. We have a lipoproteins and how they communicate with EVs. Oh, what a mess. But I think still that the approach is needed. What you are studying, I think it's great. One now comes for the genetic background. How about collaborating with biobanks, who are also taking a longitudinal samples to get access to the intra-individual variation?
No, I mean, this would be amazing.
Yes, let's continue discussion. But honestly, I think we have to take that into account. And we have been discussing a lot with Pia about all these impacts of genetic variation that may have also impact of like a standard level of certain vesicles produced by blood cells, and then this intra-individual reference. I like the idea.
Yeah, it's with the genetics. I know that. I mean, I think that there are some studies that will hopefully come out soon. We are also trying to kind of like do that, but I think the thing is that it's very hard. It's a complex genome, right? There's so many different SNPs, so many possibilities. You know, where do you start looking? And there are these big biobanks, like the UK biobank, where they have data on all of the polymorphism, but then they have also data on, you know, everything like disease and I mean, you could tap into that, but the issue is that probably it's not EV compatible. So you know, that's the problem. So yeah, we can discuss later and during the workshop, but it's something that I think we have to find a way how to approach this.
I think we should do international study.
I will. We are later.
Just a very quick question. Great talk. Why do you think you see a difference between PAC1 and CD62P?
I just don't see. Okay,
yeah,
sorry.
Why do you think you see a difference between PAC1 and CD62P?
Yeah, I know it's a complex thing. So we've tried to. I mean, we don't have any data to understand what's happening. So the thing is that it's supposedly not binding of antibodies. So what we think is that one theory that we had was that. These thrombocytes that are so there are differences in thrombocytes, and the ones that have this CD62P on their surface, that they are more, when they are active, they release more of these PS positive EVs, and these PS positive EVs are taken up. This is a signal for eat me, so maybe they are more taken up by the surrounding cells. So that this could mostly be the, let's say, the result of uptake from the surrounding cells. But we don't understand. But we see this, yeah.